Hello, can somebody confirm you can hear me?
We can hear you.
Thank you. Did not Zoom open. What should open? Yeah, there it is. Okay, any questions before we begin? Should be seeing my screen. Oh, all right, I'm not hearing anything, so let me state that today is September 25th. Boy, that's late in September. We'll be talking about Chapter 1 and Chapter 10, the microbial world in you, classification in Chapter 10. And that's it for the lecture. Any questions? We remind you, you need to have 2 lab assignments due this Friday at 8 a.m., meaning turn them in by Friday at 8 a.m., or you will be dropped from the class. There is an environmental quiz due, actually not Friday. This is going to be Saturday. I need to change that. And the online plagiarism quiz due this Saturday. You must achieve 100% on the plagiarism quiz, or you will get 0 points out of 100 on the infectious disease project. All right, any question about any of that? If not, let's go to the lesson.
Some reason on my Canvas, it's showing that I have two plagie quizzes. One of them is out of 20 and then one of them is out of 19. Do we have to do both of them or just the one that's out of 19?
Just the one I have a link to in the Canvas webpage. There shouldn't be two quizzes in the plagie quizzes in the grade books. Um,
I had the same issue where it was showing two of them, and I
yeah,
accidentally did both. I sent you an email about it, but um, it disappeared for me shortly after, and
okay.
only ended up doing the one with 19.
All right, you only need to do the one with 19 then, and that's what this link goes to. Yeah, 19 points. And the other one was something that carried over from last fall, so fall 2024, and it's not on this page, so I didn't know it was in the grade book. And it's now removed from the grade book. At least it should be. All right, let's go to the lesson. Anything else I need to talk about on that? Remember, you do have a lecture worksheet, one which is due this Saturday. I have updated this, by the way. Hopefully, it didn't change any of the question numbers. If it did, don't worry about it. I won't give you a penalty, but you should be using the questions in the order they are, answering the questions in the order they are. In the worksheet, you only need to answer one question. And what I did was I uploaded it to add more questions. And I thought I had done that earlier, and apparently I had not. All right, let's go to this. See if that picks on. I have that we had talked about where everywhere is, where we can find microbes. Oops, didn't mean to get that one there. My computer's a little slow today. I thought we had mentioned that microbes are numerically the most abundant life form on the planet. Have I talked about this slide? How many microbes there are in a gram of soil? Have I talked about this?
No,
Anyone?
I don't believe you
No.
have.
Okay, all right. In a gram of soil, there's about 1 times 10 to the eighth microbes. We're talking about prokaryote cells, bacteria and archaea. It does depend on how wet the soil is, how moist the soil is, how much organic matter is in the soil. Like, for example, in the soil of Western Washington, Western Oregon, there's probably, and especially in the forest, there's probably more microbes than there would be in the soil in, oh, I don't know, let's say, well, it's sort of a bad picture, but let's say North Dakota, just because there's more moisture in our soil, not necessarily more organics, although in the forest of Western Oregon, Washington, there probably is more organics in the soil as well. And certainly more microbes than in the soil of the sands of the Sahara Desert, for example. So it does depend on the moisture and the amount of organic matter. But about 1 times 10 to the eighth is the average. So that's average wet soil, which probably is closer to North Dakota than it is to Western Oregon, Western Washington, but you're getting the picture. Um, how does that compare to the number of microbes in a milliliter of ocean water? 1 milliliter of ocean water is about 1 gram of ocean water. Would there be more or less microbes than a gram of soil?
More.
Anyone else have a guess? All right, more is not correct. There's actually less. The oceans are a desert on the planet Earth, so there's less microbes in the ocean water than there would be in the soil. How about in 1 gram of fresh human feces? Would there be more microbes or less microbes than
More.
in the soil? Yeah, there will be more. And on one human being, well, it's much more than 1 gram to make one human being, so there's much more on a human. And on the entire Earth, there's obviously more than in 1 gram of soil. So let's take a look at the numbers here. In the ocean water, there's only about 1 times 10 to the 6 microbes per 1 milliliter or 1 cubic centimeter, or essentially 1 gram of ocean water, which is less than in the soil, about 1 times 10 to the eighth. Oops, excuse me, where am I? Right here. In the deserts of the Sahara, in 1 gram of soil, we actually only have about 1 times 10 to the 6 microbes per gram of Sahara soil. So that's about the same as the ocean water. And I did mention the oceans are a desert on the planet Earth. They have very little life form compared to, obviously, the Sahara Desert. Human feces, if it's a fresh human feces, we have about 1 times 10 to the 11th microbes in the human feces. And actually 55% of the wet weight of human feces is from microbes, the weight of microbes. Obviously, if you were to put your feces in the desert and then let it dry out and then come back sometime later and count the number of microbes in it, it'd be down to about 1 times 10 to the 6 microbes. So it does depend on how much water is in the feces, and there's usually a lot of organic matter in human feces. On one human, we have about 3.8 times 10 to the 13th microbes. On the entire Earth, we have 1 nonillion number of prokaryote cells. That's 1 followed by 30 zeros. I doubt you've ever heard of a nonillion before, but it's 1 times 10 to the 30th power. For comparison, in the human population on the planet Earth, it's a little less than 8 billion, but around 8 billion. Any question about any of that? All right. So astounding numbers of microbes. When we're talking about microorganisms, we can distinguish between a non-pathogen and a pathogenic microbe. A pathogenic microbe is one that causes disease. Specifically, in this class, we'll use it to mean human disease. Most microorganisms do not cause disease. Only a small percentage of microbes are pathogenic or disease causing. Any question about any of that? Some microorganisms actually benefit the humans. We'll talk a little bit about them. When we're talking about disease, it's important to distinguish between an infectious disease and an inherited disease. An infectious disease is a disease caused by a microbe, or I guess we could say a parasitic worm, and we already mentioned that parasitic worms fall into the category of a microbe because the infectious stages of the worm are microscopic. What's an example of an infectious disease? Can someone give me an example?
Pseudomonas. Oh,
Say
sorry.
again. I'm sorry, there's two of you speaking, so I didn't.
Sorry. You can go first.
COVID.
COVID-19 is an infectious disease caused by the virus COVID-19. What's another example?
Pseudomonas.
Pneumonia, caused by usually a bacteria or a virus getting in the lungs, or it could be even a fungus getting in the lungs. Realize there are different types of pneumonia, and they'll have different species causing them. Pneumonia is just any disease of the lungs, really. I guess disease of the lungs, which is infectious. All right. This differs from an inherited disease. This is a disease caused by someone's genes, meaning you're born with an inherited disease. What's an example of an inherited disease?
Alzheimer's?
That may not always be the case. You could get Alzheimer's. In fact, I don't think we really know what causes Alzheimer's, but uh, Alzheimer's probably is both caused by an individual's genetic makeup and the environment they are exposed to. So Alzheimer's isn't a real good example just because it's caused by both. Probably. And like I said, we don't really know the cause of Alzheimer's, but probably it's caused by both. So what's the better disease which is caused by your genes?
Type 1 diabetes.
Type 1
HIV.
diabetes, assuming that it runs in your family, yes. Somebody else was going to say something?
Asthma, or sorry, not asthma, heart disease.
Heart disease could be caused by the environment, like if you're constantly eating saturated fat or a high meat diet or something like that. So the environment can cause heart disease, as well as, I guess, smokers and maybe drinkers of alcohol probably have an increased risk of heart disease. But
What about some different kinds of cancers?
there are some cancers which run in families, in which case it would be an inherited disease. Like some families tend to get breast cancer and some get another type. But there are cancers which are not inherited. In fact, probably most cancers are either both or caused by the environment. So there are some, but not all. But you did state, somebody did state one. What the heck was it? Uh, I don't remember. Let me just say that somebody born with sickle cell anemia, they're getting it because of their genes. They have two genes which cause their cells to sickle, giving them sickle cell anemia. And that would be a case of an inherited disease. Okay, that's the classic example. Any case about that. Somebody did mention one. I'm trying to remember what it was, and I just don't remember now. So um, obviously some cancers do run in families, in which case that would be an inherited disease. All right, any question about infectious versus inherited disease? An infectious disease is obviously one that spreads. from one patient to another caused by a microbe, which is why it can spread. An inherited disease, they do not spread. You are born with an inherited disease. Here are some pictures of some microorganisms. Let me blow this up. A bacteria. And that's about the average size of a bacteria, around 1 micrometer, maybe up to 2 micrometers. That's a little bit larger. That's a little larger, about 3 micrometers. Some of them are small. This is 1 micrometer. This is actually a large microbe because that's 50 micrometers. This is some fungus. That looks like to me like a red blood cell, but that would be a microscopic cell. And there is actually a colonial microbe. And you see each of these dots here? One of those dots is a cell. And so this is the parent colony. This is Volvox. And it has made 6 offspring colonies. And the parent will open up, and then these offspring colonies will come out, and then they'll go separately and form their own colony. This is a microbe, 50 micrometers here. So this is what, around 150, maybe 200 micrometers across. You need a microscope to see it, but it is colonial. Most microbes are single-celled, like this bacteria here. And then another example of a microbe, each of these red dots is HIV, and it is considered a microbe. This is actually a T4 cell, CD4 lymphocyte. So viruses are smaller than bacteria. Oh, well, that's 50 micrometers, but that's much less than 50 micrometers. Microorganisms play numerous beneficial roles on the planet Earth and for humans. Here are some examples of some of the beneficial roles that microbes engage in. Microbes are responsible for decomposing organic waste. The only exception to that is some mushrooms can decompose organic waste in addition to microbes. And usually microbes are always doing it, but I suppose under a certain environment, you might have only the mushrooms growing. And that would be very unlikely. You always have the microbes with the mushrooms. Our producers, microbes are producers in the ecosystem, meaning some microbes are photosynthetic, producing both the sugar for other organisms, including humans in the ecosystem, and the photosynthetic organisms tend to also make oxygen. Not all photosynthetic organisms do make oxygen, but many microbes are photosynthetic, produce sugar in the ecosystem as well as oxygen. And then some microbes are also producers, but they are chemosynthetic. The chemosynthetic bacteria using chemicals to get their energy needs rather than light to get their energy needs. And we'll talk a little bit about both of these later in the course. I don't know if we'll talk about them today, probably not, probably in a later lesson. Some microbes are involved in the nitrogen cycle. We already talked about that. Many microbes produce substances or chemicals for human use or use in the environment. Like microbes make industrial chemicals, such as ethanol and acetone, which humans can use to make ethanol and acetone. Some microbes make insecticides, which can be used against insects to help protect crops. Some microbes ferment foods, like the making of vinegar, cheese, bread, beer. All of these products are made because of microbes. We would not get leavened bread without the use of a microorganism, specifically yeast, sometimes yeast and bacteria, like when you're making sourdough bread. We use products used in manufacturing by using microbes, like the making of cellulase. We use the microbes to make the cellulase and then use the cellulase. Antibiotics. Some microbes make antibiotics, which then humans use. For example, penicillin G is made by a microbe. And then there are other products used in medicine which microbes make. For example, human insulin is actually made by bacteria. What they did was they took the human gene out of the human for making insulin and then put it into a bacteria, had the bacteria make the insulin, and then purify it out, and then make it available for the diabetic. My father was a diabetic, first became a diabetic in the early 1960s, I believe, and he first was using human insulin. It was very expensive because they had to purify it from human blood. And then they started using sheep's insulin because it was cheaper and easy to purify from sheep's blood. And then later on, they started using bacterial uh, insulin, meaning they put the human gene into bacteria and grew up the insulin and then purified it that way. Any question about any of that? So microbes play beneficial roles. Biotechnology is the use of microbes and other organisms to produce foods and products for human use. Biotechnology is actually centuries old. An example of biotechnology would be using microbes to make beer or wine and using microbes to make, what do you call that? Sauerkraut. Centuries old. And that's biotechnology. Most people think of biotechnology as something new and modern. That's actually genetic engineering, a new technique of biotechnology, where we usually take a gene out of an organism, such as a human, and then put it into another organism to get that other organism, such as a microbe, to make the product of the gene. There are applications of genetic engineering, including producing a variety of proteins from bacteria and fungi. Like we can make antibiotics from bacteria and fungi by taking the gene out of an organism and putting it into a bacteria or a fungi. Got another example of that. Making vaccines. We can take proteins out of a human, put them into bacteria, grow that up, and make a vaccine or an enzyme from that bacteria. You can use genetic engineering to start gene therapy. A child is born with a defective gene, such as a defective immune system. You can take the correct gene out of a functioning human and put that gene into actually they put it into a virus, which is a microbe, and then infect the virus into the child's immune cell, grow up the immune cell in culture, and then inject those immune cells into the patient. The patient then has a functioning immune system for several months. It's only for several months because the lymphocytes or the immune cells injected into the patient don't live forever. But that's an example of gene therapy where we're taking genes out of an individual, putting them into cells, and then putting them into the patient to get the therapy. And then there's genetically modified bacteria where we can modify crops to protect them from insects, like putting in an insecticide into the crop species itself. Or there's a gene that stops freezing, meaning it puts in an antifreeze. You can put it into the crop and then protect the crop from freezing. Genetically modified bacteria. I think you're all familiar with the genetically modified tomato, the flavor savor tomato, which actually was very popular for a while. You could find it in almost every supermarket. Apparently, people didn't buy it, so it's very hard to find the flavor savor tomato. The stores loved it because you could pick the tomato and then it would stay ripe for a long period of time before spoiling. And people didn't buy it, and so it's not very common anymore. Anyone not ever heard of the flavor savor tomato? All right. It was a genetically modified tomato. I'm sure you've heard of genetically modified crops. That was one of the first. Microbes are important to human welfare. There's a microbial ecology with every human, where microbes play an important part in the ecology. Microbes are important in the ecosystem for recycling carbon, recycling nitrogen, recycling sulfur, recycling phosphorus in the ecosystem. And microbes are extremely important for recycling nutrients in the ecosystem. Like I stated, nutrients are recycled only by microbes or by mushrooms, fungi, which most fungi are microbes, but mushrooms are not. And then when these nutrients are recycled, they can be used by plants and animals. So when an organism dies, having carbon, nitrogen, nutrients, sulfur, and phosphorus, the microbes can recycle those nutrients and make them available for other, excuse me, for other plants and animals. There are examples of microbial-mediated biochemical cycles that support life on Earth, and this was first discovered by Dr. Winogradsky, and Dr. Bejerink. They were the first to show the recycling of nutrients by bacteria in the ecosystem. And the reason why they were able to find this is because Winogradsky, when he was studying microbes, he didn't study a pure culture of one species. He would study microbes growing together in an ecosystem, meaning many species growing together. And because there are many species growing together, he was able to see the the nitrogen cycle or the sulfur cycle. I know he did those two. He might have done the carbon cycle as well. Oops, come on, stop it. I need to shut that down when I'm not using it, but I don't know how to do that. So, any question about any of that? If Dr. Winogradsky was only studying one species, he would not have been able to find the nitrogen cycle, because if you remember, let's go to near the very beginning. I'm on slide 20. There are different bacteria and archaea that do each step. Like the bacteria that make nitrite are different from the bacteria that make nitrate. And then that's different from the bacteria that change nitrate into nitrogen gas. So you need an ecosystem of microbes to be able to see the nitrogen cycle. And that's why Dr. Winogradsky was able to find it, because he was using an ecosystem when he was studying microbes. Any question about any of that? All right. Microbes are important for human welfare, such as bioremediation. Bioremediation is literally to remedy life, or life and to remedy, such as spraying this oil spill with microbes to have the microbes digest the oil, breaking it down into CO2 and water, solving the environmental problem. So bioremediation, using living organisms to deal with an environmental problem, which is exactly what these two gentlemen are doing. We use bioremediation in our sewage treatment plants to break down the sewage. You can also use bacteria to break down various pollutants like the oil spill here, the oil. And you can use bacteria to detoxify mercury. Elemental mercury, meaning regular plain mercury, is toxic, very toxic to humans. But if you get the bacteria, you don't break down the mercury, it's still an element, but you can make an organic form of the mercury, which is less toxic than elemental mercury. And that would be bioremediation to make the mercury less toxic. It still is toxic because it has mercury, but it's less toxic. Any questions about any of this? I mentioned that you can use microbes to form biological insecticides. It's an alternative to a chemical pesticides. You can use the microbes or the insecticide made by microbes to prevent insect damage from crops, as well as prevent disease transmission. An example is Bacillus thuringiensis. This bacteria is actually fatal to many insects, but the bacteria is harmless to humans and animals and plants. And way back in the 1960s, my father bought some Bacillus thuringiensis and put it on the apple trees to prevent the apples from getting worms. So way back in the 1960s, we had Bacillus thuringiensis available. The trouble was my father was too lazy to put it on the tree more than once, and the Bacillus thuringiensis should be put on the tree every 2 weeks to prevent the worms, as well as every time after a rainfall, because the rain tends to wash the Bacillus thuringiensis bacteria off the apples. And he was growing his apples in Western Oregon, which, as you know, gets a lot of rain. And so obviously our apples got worms. And I don't know why didn't read the directions because it told them what to do, but he only applied it one time, and he knew with insecticides, if you spray the apples one time, that's not going to prevent the apples from getting worms. The Bacillus thuringiensis used one time did reduce the number of worms, but you have to apply it every 2 weeks, which is about the same as you have to apply a man-made insecticide on the apples to prevent the apples from getting worms. So you need to repeat every 2 weeks. And then with the case of Bacillus thuringiensis, you need to also repeat after the rain. Any question about any of that? All right. Any question on anything so far? If not, we're going to change gears a little bit and talk about the naming and classifying of microorganisms. Both the scientific naming and the scientific classification was begun in science by one individual shown here who did it in 1735. You don't need to know the year there, the 18th century. Does anyone know who I'm talking about? Who this is? Beginning the scientific naming and classifying of all organisms?
Is it, I'm gonna butcher it, but Carolus Leninius?
That's good. Yes, he liked he liked Latin so much, he even changed his name, which strictly speaking was something like Carl Linné. I think he was Swedish, and he switched it to Carolus Linnaeus just because he liked Latin. And he established the scientific naming system as well as the classification system. And the naming system, also called the scientific nomenclature, He gave each species two names, a genus name and a specific epithet name. So a species name has both the genus and the specific epithet. Like for humans, our scientific name is Homo sapiens. Homo being the genus and the specific epithet being sapiens. It's called the scientific nomenclature or binomial nomenclature because each species has two names. Bi means two, nomial means names, so two names, naming system. He also began the classification system, and we still use his scientific nomenclature system because it was so good, we still use it today. We no longer use his classifying system because he classified all organisms into either plants or animals. And as you know, a microbe is neither a plant or an animal. There are different things. A fungi is not a plant or an animal either. But he classified everything into plants or animals. We don't use that anymore. We'll talk about what we use now in a little bit. The binomial nomenclature system uses names in either italicized or if you can't italicize, you underline it. Like if I were to go and write up Homo sapiens on the blackboard, which I guess I can show you a whiteboard here, but and I can't italicize it. If I write it, I then underline it to mean that it is a scientific name. The genus name is always capitalized. The specific epithet is always lowercase. H in homo is capitalized. S in sapiens is always lowercase. The scientific nomenclature, all the names are Latinized and they're used worldwide. That means that whatever language you speak, if you want to describe humans, you can say Homo sapien, and that will be understood in whatever language you're in, whatever country you're in, whatever language they use. Homo sapien is used worldwide to describe the human species. And then Staphylococcus aureus is used worldwide to describe the species Staphylococcus aureus. The name may be descriptive or it may honor a scientist. For example, Homo sapien is descriptive. Homo is the genus of individuals who walk on 2 feet. Sapiens means wise. So this means wise man that walks on 2 feet. Escherichia coli is both descriptive and honoring a scientist. Coli is named that because it can grow in the colon. It grows in the intestine of humans. And then it's named after Dr. Esther, an early microbiologist. Staphylococcus aureus is descriptive. Staphylococcus, coccus means it's a ball-like cell. Staphylo means it grows in a clump like grapes. So Staphylococcus actually means growing like grapes. And then aureus means golden, and it's a golden cell growing in a clump like a grape clump. Clump of grapes, I guess. Any question about any of that? After first use, scientific names may be abbreviated with the first letter of the genus and the specific epithet spelled out. So the first time you should use Staphylococcus aureus and Escherichia, sorry, I'm having trouble saying that there, coli are found on the human body. And then after that, you can say S. aureus and E. coli is found on the human body, and E. coli is found inside the human body in the intestinal system. Any question about any of that? Now, with some names, like E. coli, it is so commonly used and so well known that even scientists tend to use the term E. coli rather than say Escherichia coli the first time. They should, they don't always, but they should spell it out the first time and then abbreviate it. And this is just an exception because E. coli is so commonly known. All right, that's the naming system. I mentioned that both the naming and the classification system was started by Carolus Linnaeus. I think your textbook calls him Carl Linnaeus, like it Latinized one name but not the other. And like I said, you probably should use Carolus Linnaeus or Carl Linnaeus. That was his name before he Latinized it. I forgot to mention the textbook sort of does an in between. But let's talk a little bit about the classifying of microorganisms. It too was begun by Carolus Linnaeus. We don't use his classification system anymore because, well, it wasn't appropriate. Not all organisms were plants or animals. So when we look at all organisms on the planet Earth, we first separate them down into the three-domain system. And that is the domain bacteria, the domain archaea, and the domain eukarya. The domain system, also called the tree of life, because this looks like a three-branching tree or a three-trunk tree, and then it branches as tree branches do. This is based on the genetic data of a species. So if we were to find a new species and to determine which domain it goes into, all you need to do is sequence the species DNA. And then its DNA will be more similar to its domain. Like if we found a new species of bacteria and we sequence it, we would see that its DNA is more similar to other members of bacteria than it is to other species in other domains. So the domain system is based on the genetic data, the genetic similarity or the DNA sequence of the organism. And the more similar it is, the more similar it will be, meaning the more closely related it will be. And that's why it will be more similar to, if it's in the domain bacteria, any species in the domain bacteria, than to any species outside the domain bacteria. There are three domains: domain bacteria, domain archaea, and domain eukarya. And they each are more similar to other species in their sequence of DNA than to any species outside the domain. Any question about any of that? All right. You can divide the classification system down from the domain. We're only going to talk about one, but let me see if I can remember that. There's kingdom, phyla, class, order, family, genus, species, and then subspecies. We're only going to talk about the grouping after the domains, and that is you can separate the domains into kingdoms. The number of kingdoms there are is currently in debate, and this debate has been going on for about 20 years. I don't think scientists will ever agree on the number of kingdoms there are. We're going to use the six-kingdom approach, which means that there's a kingdom bacteria and a domain bacteria. All of the domain bacteria fall into the kingdom bacteria, so the kingdom and the domain are the same. And the same with archaea. The kingdom and the domain are the same. For the domain eukarya, we're going to separate this domain into four different kingdoms. There's kingdom plantae, the plants, kingdom animalia, the animals, kingdom fungi, the fungus, and kingdom protista, the protists. Kingdom plants, animals, and fungi are monophyletic groupings, meaning all the plants will be more similar to any other plant than they are to an animal, a fungi, or a protist. But the kingdom Protista is not a monophyletic group. A protist, or kingdom Protista, is something belonging to the domain Eukarya, which is not a plant, not an animal, not a fungi. So the kingdom Protista was always a collection of other eukaryotes that was not a plant, animal, or fungi. It's not a monophyletic group. Any question about any of that? All right. Now, there is no definition for what a kingdom is. Like for the domain, the definition is your DNA sequence will be more similar to other members in your domain. So we have a definition for what a domain is. For the kingdom, we do not have a definition. What's a plant, what's an animal, what's a fungi, and what's a protista? And different people will argue about what kingdom a organism belongs to. Like some people will say that the kelp that washes up on the Oregon beach, they'll argue it's a plant. It's photosynthetic, multicellular. It's a plant. Others will argue, no, it's a protist. I'm not going to argue why it's a protist, but and like I said, there's no definition, so you can disagree. And another thing is the number of kingdoms is in debate. Some people say there are 3 kingdoms, the same as the domains. Some people say there are 5 kingdoms. Here's 4, and then they lump those 2 together as a kingdom. Most people say there's 6 kingdoms, what I've shown here. And this was the system in use before scientists started arguing about how many kingdoms there were. Other people say there's 9 kingdoms. If there's 9, what you do is you divide the protist into different kingdoms. So whatever needs, you get 9 kingdoms. So what is there? I don't know. And then they say it's 15 kingdoms. All they're doing is dividing the protist into more kingdoms. With the 20 kingdom approach or higher order, there's 30 kingdom approach, 50, 55 kingdom approach. With the higher order kingdoms, you start dividing the bacteria, the archaea, and some of the eukarya, like the plants, can be divided into 2 different kingdoms in the higher orders. I think it's like 30 kingdom approach. You can start dividing the plants into 2 kingdoms. Any question about any of that? So we're using the 6 kingdom approach just because it's simple. It's the last approach biologists used, but the number of kingdoms is currently in debate, and we're not going to try and solve that debate because they've been debating it for about 20 years now. All right. Your textbook talks about a cell type. It says that the bacteria have prokaryotic cells, the cell type being prokaryote. Archaea also have prokaryotic cells, the cell type being prokaryotes. The eukarya have eukaryote cells, the cell type being eukaryotes. This is the way your textbook talks about it, so I'll use that way. But some people call these cell types a superkingdom. if you look at the classification system, you have all living things. And then before you hit the three domain system, you have the super kingdom, being prokaryotes and eukaryotes. And then we split the prokaryotes down into the domains. So there's three domains. The super kingdom name was around before the domains. So the domains started in the 1960s, I think the late 1960s, when people started sequencing species and discovered that there were three domains. The super kingdom was around before that. People knew about cell type prokaryotes and cell type eukaryotes. And so you call it a super kingdom. It doesn't matter what you use. You can use what the textbook uses or a super kingdom approach. Let's talk a little bit about the domain bacteria. They are prokaryotes, their cell type. The bacteria have peptidoglycan in their cell wall. Most of the bacteria multiply by binary fission, where one cell divides into two, splitting evenly. Bacteria can use almost all energy sources. Bacteria can get their energy needs from an organic chemical such as glucose, and that's what we get our energy needs from, an organic chemical such as glucose. But bacteria, especially different species, can get their energy needs from an inorganic chemical such as iron or hydrogen sulfide gas or sulfate or nitrate. The point is they get their energy needs from an inorganic chemical. And some bacteria can get their energy needs from photosynthesis, meaning they can harness the energy in light for their own energy needs. So bacteria, depending on the species, can get their energy needs from really all the energy sources that living things can use. Most bacteria are free living, either decomposers or primary producers, but some bacteria are parasitic and some bacteria are pathogenic. Remember, pathogenic is it can cause disease, and we're talking in this class about human disease. So some bacteria are human pathogens. Any question about the domain bacteria?
What is the peptidoglycan?
We'll talk about it later. It's a chemical in the cell wall of bacteria. It's the most common makeup of the cell wall of bacteria.
Okay.
So the most common molecule in the cell wall of bacteria. We'll talk about it later. All right. Let's talk about some of the characteristics of archaea, also called archaebacteria. Their cell type is prokaryote. Does anybody know, do the archaea have peptidoglycan in their cell walls?
They do not.
Yes, you're correct. Archaea do not have peptidoglycan in their cell walls. Peptidoglycan is only found in the domain bacteria. Some of the archaea are found in extreme environments, and this is where we first found the archaea, in extreme environments, including methanogens, extreme halophiles, that means extreme salt environments, extreme thermophiles, meaning extreme temperature environments, hot temperatures. But when we look at all the Earth's biomes, we find the archaea in every biome. So we can find it everywhere on the planet Earth. It is a significant portion of the bacteria in the soil and the bacteria in the plankton. So it is significant, at least in those two environments. Does anybody know, are any of the archaea, any species, are any of them pathogenic? You can always take a guess. Worst you'll be is wrong.
They are not pathogenic?
You're correct. They are not pathogenic. There is no known archaea which causes human disease. For this reason, other than this lesson here, talking about the archaea, I am not going to be talking much about the archaea in this class. It's not that I don't think they're important, not that I don't think they're interesting. It's just that they do not cause human disease. And the focus of this class is on the bacteria which cause human disease. So we're not going to talk about the archaea very much in this class. Any question about any of that? All right, let's move on to the domain Eukarya. There are 4 kingdoms in the domain Eukarya, at least in the system we're using. Hopefully you didn't see that. The domain Plantae, the kingdom Plantae, the kingdom Animalia, the kingdom Fungi, and the kingdom Protista. All of the kingdoms and all of the eukaryotes lack peptidoglycan. Peptidoglycan is only found in domain bacteria. Let's talk a little bit about the kingdom fungi. Fungi is plural, singular fungus. Like if I pick up one mushroom, that is a fungus. Any question about any of that? You don't really need to know about their cell walls. You should know that fungi use organic chemicals for their energy needs, something like glucose, which is very similar to us. There are multicellular molds and mushrooms, which are multicellular. They consist of masses of mycelia, shown here, which are composed of filaments called hyphae. And then there are unicellular yeasts, shown here. Most fungi are decomposers, but a few fungi are pathogens, like Candida albicans can cause skin disease as well as nail disease, eye disease, thrush, which is a disease of the tongue or mouth, a few other diseases, jock itch, yeast infection. We'll talk very little about Candida albicans later in the term, but it is a fungi which can cause human disease. It is also a part of your normal flora living on your skin. And I shouldn't use the term flora, I should say part of your normal microbiome living on your skin. But it can be a human pathogen. Okay, that's it for the kingdom fungi. Let's talk about the kingdom Protista, the protists. Some of the protists are plant-like protists. We call them algae. Some of the protozoa are animal-like protists, and we call them protozoa. And some of the protists are fungus-like protists. And we only call them fungus-like protists. The algae are all plant-like protists, meaning they have cellulose in their cell walls. They use photosynthesis for obtaining their energy, and they do produce molecular oxygen and glucose and other organic compounds, which humans could eat. Algae isn't very tasty, but you could eat it. All right, any question about the algae? The protozoa, also called the animal-like protists, include the amoeba, the paramecium, the euglena. Some of them are modal, and if they're modal, they may move by flagella, or they may move by cilia, or they may move by pseudopods. A pseudopod is where you put out the cytoplasm and pull it up in the opposite direction, and then the cell can move that way. Some of the protozoa are parasites, like some of the paramecium, some of the amoeba, none of the euglena that I'm aware of. But some of the paramecium and the amoeba can cause human disease, like amoebic dysentery caused by Entomoeba histolytica. An amoeba, you don't really need to know that, but just give you an example. The fungus-like protists, we're not going to talk much about them. There's only one that is commonly known, and that is the cause of the potato blight is caused by a fungus-like protist. And if you don't know, it was the fungus blight that killed the potato crop in Ireland, which brought a whole bunch of Irish into the United States. I think it was the late 1800s, maybe the early 1900s, from the blight of the potato blight. Any question about any of that? All right, let's talk about the kingdom Plantae. This includes the mosses, the ferns, the conifers, and the flowering plants. All of kingdom plants is multicellular. There is no single-celled plant. And just to talk about this, the protozoa are single-celled. The fungus-like protists are single-celled. The algae, some of them are single-celled, some of them are multicellular, like the Volvox is multicellular. So all the plants are multicellular. There's no plant that causes human disease. So that's it. That's all I'm going to talk about kingdom plants. Kingdom Animalia, the animals, can be separated into the sponges, the worms, called helminths, the insects, and the vertebrates. Humans are a vertebrate, by the way. All of the animals are multicellular. Some of the animals can cause human disease. Specifically, some of the worms, some of the helminths, can cause human disease. And so we'll talk a little bit about them in this term later on, not really today. We're not really going to talk about the sponges, the vertebrates, and really not much about the insects either. Any question about the kingdom Animalia? All right. What comes under the umbrella of microorganisms includes organisms that can cause disease, that's bacteria, archaea, virus, and prions. Remember that virus and prions are special because they're not organisms, they're not cells, they are a microbe, they do cause disease. Certain of the eukarya, the protozoa, the molds, and the yeast. Some of the algae, which do not cause human disease, are microbes, but not all of the algae are microbes. Like the kelp washing up on the Washington beach is not a microbe. And then the helminths, the worms. Even though the worms, the adult worms, are not microscopic, we've talked about this before, and that the infective stage, as well as some of the life stages of the worms, are microscopic. For most worms, the infective stage is the fertilized egg, but there are a few microscopic worms that can burrow into the human skin and then grow into the adult worm. Let me think about that. I used to know what that one was. That Ascaris? I don't remember. The helminths, the adult animals, are multicellular. But oftentimes, if the infective stage is a fertilized egg, that's a single cell. Many of the helminths are parasitic. The parasitic worms are flatworms and roundworms. And you should know that a flatworm and a roundworm, even though we call them worms, they are not genetically related to each other. In fact, the flatworms are as different from the roundworm as the flatworm is different from a human. So the genetic relationship between the flatworm and the roundworm is the same as the genetic relationship between the flatworm and the human. Any question about any of that? So even though we call them worms and a helminth, this is really a human, I would say, mistake, and that is the flatworms and the roundworms are not related to each other. They are genetically as distinct from each other as they are distinct from a human. Any question about any of that? All right. The viruses. Let's talk a little bit about viruses. They are acellular. They don't have a cell. They do have DNA or RNA as their core molecule or their genome. They do not have both. They have one or the other. The core molecule is surrounded by a protein coat. The coat may be enclosed in a lipid envelope. Not all viruses have a lipid envelope, but some do. And when do viruses replicate? They only replicate when the virus is in its host cell. If the virus is outside of its host cell, the virus cannot replicate. Viruses are not part of the three-domain system because they aren't really an organism. They don't have enough DNA to even be certain of their evolutionary history. They're a small molecule of DNA compared to any other species of cellular organism. Any question about viruses? It's a very brief introduction. We'll have a whole lesson on viruses later in the term, very late in the term. All right, here's looking at the domains of cellular organisms. We can separate the cells into bacteria, archaea, and eukaryotes. The bacteria and the archaea are cell type prokaryotes. The eukaryotes are cell type eukaryote. We look at the chromosome of bacteria, archaea. They're generally one and a circular chromosome, meaning I'm trying to draw a circle here. Do it over here. Meaning it's in a circle, and the place where we begin, right here, is the place where we end. Circular chromosome. The eukaryotes, their chromosomes are usually many. In fact, they're always many. There's never one chromosome in a eukaryote. And there are linear chromosome. For example, the X chromosome shown here, it has a beginning place here and an ending place here. And the ending place is different from the beginning place. There's a few other differences in the chromosome. I'll talk about them later in the term. We can look at the ribosomes of bacteria, archaea, and eukaryotes. The bacteria and the archaea have similar ribosomes. They're both 70S in size, slightly smaller than the eukaryote ribosome, which are 80S in size. We'll talk about the S later. That's a unit that biochemists have for measuring ribosomes, and it stands for the sedimentation coefficient, where the ribosome sediments in an ultracentrifuge gradient. When we look at the presence of peptidoglycan in the cells, we find peptidoglycan in the cell wall of bacteria. We do not find peptidoglycan in archaea or in eukaryotes. Any question about the three domains here? So which domain is more similar to the other domain?
Bacteria and archaea, right?
Not really. It's true they are both cell types prokaryotes, that they have one circular chromosome, they have similar ribosomes, but if you look at the peptidoglycan, they differ. In fact, the archaea are similar to the eukaryotes. In fact, let me go way back here. I meant to mention this way back when I was talking about archaea. If we look at the archaea here, let me blow that up a little bit. Maybe if I go here, get that out of the way. You can see that the archaea and the eukarya actually were together for a little bit longer than the archaea were together with the bacteria. And that means that the archaea are slightly genetically more related to the eukarya than the archaea are genetically related to the bacteria. It's very slight, okay? But the archaea are slightly more similar in their DNA sequence to the eukarya than the archaea are to the bacteria. And that's just because they had a common ancestor, which was later the common ancestor between the archaea and the eukarya than the ancestor between the archaea and the bacteria. But it's very slight. All right, let me end this, go back to where I was. Right there. All right. Let's come here. So I'm going to be switching gears again, talking about the history of microbiology. We're going to talk about a brief history of microbiology. And in the history of microbiology, it largely concerns the debate over whether spontaneous generation gave rise to life or whether life could only come from other living cells or organisms. It was never debated about where a baby came from, but certainly where microbes came from, if they could spontaneously generate. That was debated for a long period of time. We'll talk about the golden age of microbiology. That's early microbiology. Whenever microbiologists were looking at something from about 1857 to 1914, they were looking at something new, something that had never been seen or described before. And that's known as the golden age of microbiology. You don't need to know the dates of the golden age of microbiology. You just need to know it's the early age of microbiology. Here's looking at a brief summary of microbiology with the golden age of microbiology shown right there. And it does include some things that happened before microbiology was invented. In microbiology and the history of microbiology, I'm going to talk about dates just because it's easy to anchor a topic to a date and then give them in a specific order with the date. But you do not need to know any of the dates except for this first item. You need to know that the ancestors of bacteria were the first life on Earth was seen in the early rock fossils, and you can find the ancestors of bacteria all the way back to 3.5 billion years ago. Any questions about any of that? So as far as we know, the first life on Earth arose about 3.5 billion years ago, and it was an ancestor of bacteria, and that's actually a fossil right there, an actual fossil. There's another one right there. This is the artist's rendition right there, what they're seeing. And that's the only date you need to know. Any question about any of that? All right, about 4,300 BC, the Babylonians actually wrote recipes for making beer. And this is the first time we have a record where something was used which used microbes to make the beer. Now, the Babylonians could not see the microbes, so they did not know that the microbes were making the beer. But my point is, this is the first record we have of somebody using microbes to make something, beer. In about 600 AD, the Mayans wrote records making a fermented beverage from cacao that today we would call chocolate. So you don't need to know these dates here, but you probably should know the rank order that before the Mayans could make a record that they made a fermented beverage from cacao, that we're talking about chocolate, that there had to be someone who wrote down first use of microbes, and that was the Babylonians, and that had to be after the first life arose on the planet. So you should know the relative rank order of these events, but you do not need to know the dates. Any question about any of that? And it should be obvious that if the Mayans wrote a record for making, we're going to call it fermented cacao or chocolate, that somebody had to write something about microbes making something for the person, something that had to be done first, and that had to obviously be done after life arose on the planet. So if you use common sense, you'll even know the rank order. All right, the first observations of microbiology had to happen after the invention of the microscope. You don't need to know the date, but in 1590, Janssen developed the compound microscope. I don't know why he made it. It wasn't to look at microbes because he didn't know they existed. And in fact, nobody used the microscope to look at cells for almost 75 years. Robert Hooke was the first person to use a microscope to look at cells or living organisms. And when he did, he saw cells, which he called little boxes. And he's the first person who, at least we know of, wrote a record that he looked at plants. And that marked the beginning of the cell theory. The cell theory is that all living things are composed of cells. It was developed further by others like Virchow, actually wrote up the cell theory and published it much later. But in 1665, the theory began because Robert Hooke looked at plants and he found cells in them. All of this above is not strictly microbiology. The invention of the microscope is not microbiology. Robert Hooke looking at plants are not microbes. Okay? Let me talk a little bit about cells here. Oh, we're out of time. So let's put it here.
I have a question, and it may have already been answered, but are we gonna have access to this slide?
When I'm done with the lesson, you're not going to have access to it until we're done with the lesson.
Okay.
Okay. So if there's anything you want to write, go ahead and write now. I'll leave it up for a little bit. I need to talk about cells. Hopefully I'll remember. Remember to remind me I need to talk about cells. Okay. How, what he saw when he looked under the microscope and why he called it a cell. All right. Actually, that's 41. That's not bad. We got about 67, 27 more slides to do. All right. Any questions? If not, I'll say goodbye. I'll see you at 6:31 for the lab. I'll give you 10 minutes off.